Hi, everyone. I'm your host, Rob Culverhouse. I'm an associate professor in the Department of Medicine at Washington University. So welcome to the first ICTS Precision Medicine virtual session at the Venture Cafe. We plan to continue meeting on the third Thursday of the month at 4 o'clock. And the virtual sessions, as we just mentioned, will last only 45 minutes instead of the usual hour to allow for speakers for the next talk to get set up. And we're continuing to adjust to the pandemic, so I'll need to ask you to keep just checking the Venture Cafe website or the ICTS Precision Medicine website for information about what's going to happen in the upcoming meetings. Our website is just ictspm.ustal.edu. Our mission is to connect the innovative biomedical science generated at the local universities WashU, SLU, and UMSL with entrepreneurs and businesses in the St. Louis region. We hope you find these sessions will stimulate your thinking, and we encourage you to share your insights and ideas with the speakers after their presentations. We're really hoping for some synergy here. And tonight's theme is innovative approaches to chronic diseases, specifically diabetes and kidney disease. Our speakers are Doctors Jeffrey Millman and Mohamed Zayed. Jeffrey is an assistant professor of medicine and biomedical engineering at Washington University, and Mohamed is an assistant professor of vascular surgery at WashU, has a PhD in pharmacology, and is the chief medical officer for a startup focused on creating devices placed inside veins to remove blood clots. So Jeffrey is our first speaker. The major focus of his lab is the production and study of insulin-producing cells from human stem cells. Today, he'll tell us about how this technology can revolutionize treatment for diabetes through drug screening and cell replacement therapy. So take it away, Jeffrey.
Great. Let me see if I can actually do the share screen now when it actually counts. And does that look okay?
Looks great.
Great. Awesome. Well, thank you everybody for coming to this. I am the guinea pig for trying to do Venture Cafe virtually. So if we encounter any problems, I hope you are able to bear with it. I do also want to apologize up front that no, there will not be any free beer deliveries coming to your homes. I did ask and I did try to make it happen. So we'll have to do this, just with our cups of water or whatever you may be having at home. So I am a faculty in the Division of Endocrinology, Metabolism, and Lipid Research, and I run a laboratory over at the medical campus that is focused on using stem cells for the study and treatment of diabetes. So the team that I have over there is a mixture of people from a bioengineering background and various types of biology backgrounds, including cellular biology. And so the reason why we are using stem cells in this context is, I don't know if we can mute everybody because there's a lot of feedback coming now. Rob, can you try to, I guess it's gone now. Thank you. Is that is this major problem of diabetes. So diabetes is caused by the death or dysfunction of the insulin-producing beta cell, which is a very specialized cell type found within the pancreas. And it's really this insufficient number of healthy beta cells that gives rise to all forms of diabetes, including type 1 diabetes, type 2 diabetes, and completely genetic forms of diabetes. And the schematic here, of course, really simplifies, overly simplifies how diabetes is managed, but it can essentially be thought of as patients taking exogenous insulin or other sorts of drugs in order to control their blood sugar levels. And obviously, insulin is a life-saving drug for many, many people. But essentially, what this boils down to is that the patient is trying to basically do the job the beta cell is supposed to be doing, and that is injecting themselves with insulin or forcing beta cells to produce more insulin when their blood sugars are high. A beta cell can naturally do this, and it's impossible for a individual to constantly be monitoring their sugar and constantly being able to inject themselves with the appropriate dose of insulin. And so what this means in many situations of diabetes management is that blood glucose, blood sugar levels are not controlled precisely. And again, this is even though patients are having to measure themselves multiple times a day and inject themselves multiple times per day. And so this imprecision in the control of their blood sugar levels gives rise to many of the associated health problems with diabetes. So instead of having the patient do the job of the beta cell, at least conceptually straightforward solution to this would be to just give the patient more healthy beta cells. And if this was successful, this would essentially be a functional cure for their diabetes. So diabetes is a very common disease with hundreds of millions of people worldwide suffering from it, and this number is only expected to continue to rise with time. So what the challenge that this presents to us is that we need a large-scale sustainable source of replacement cells and tissues in order to even begin to chip away at this large number by making new cellular therapies available as an option for these patients. And so this is really where my lab is focused on here, is that in the lab we are generating beta cells from stem cells using approaches that we developed that essentially try to recapitulate the signals the beta cells see during development normally, but instead of doing this in the embryo, we are doing it in the lab. And we already worked out many of the technical hurdles associated with doing this to be able to generate billions of cells fairly easily in an academic lab that's not set up for large-scale manufacturing by any stretch of the imagination, at least not on an industrial scale. And inside of these cell culture vessels, whether they be hyperflask or bioreactors, we are able to grow these beta cells in clusters of a few thousand cells. And this is just a cute picture showing some genetically engineered cells that glow red when they are producing insulin. And so taking one of these clusters that are about the same width as a hair on your head, taking a cross section of that, and we can stain to see that these cells are expressing markers of beta cells. And this includes there being many of the cells having insulin inside of them. But these are not just simply factories of insulin. These cells are able to, in a very well-regulated manner, control the release of insulin. As the data over here shows, that at low levels of sugar, you have kind of a low baseline level of insulin secretion, going to higher and even higher levels of insulin or higher levels of sugar, the insulin secretion correspondingly is able to increase. And it's really this well-regulated secretion that makes these cells special. We, of course, could just have insulin in a sponge or biomaterial and have it be kind of a time-release sort of thing, which is sometimes used in the veterinary field, actually. But in order to have really well-regulated control of blood sugar levels inside of a person, we really need to have the cells being controlled by sugar levels in terms of their insulin secretion. And here's one such example of that in mice that were able to make diabetic with a drug that destroys the mouse's beta cells. If we don't do anything to the mice, they just stay diabetic for the observation period. Transplantation with our cells is able to reverse this severe hyperglycemia in about a week. So in addition to the cells themselves being a potential therapeutic, these cells can also be used to, these lab-grown cells can be used to recapitulate disease. Just one simple example here on the screen, again, showing the beta cells at low and high sugar levels will go from low to high levels of insulin secretion. We can give them, in this case, we give them a compound that mimics the stress of being in a diabetic environment, and we're able to completely ablate their ability to mount a elevated insulin secretion in response to high glucose. And so we've been spending many years now defining chemical and genetic mimics of the diabetic state, and have developed functional and transcriptional readouts of how these cells are able to respond to that. We've also made some genetically engineered readouts that help to even further increase the compatibility of these cells with high throughput screening to hopefully find new drugs. So this is essentially what the academic mission of my lab has been. We've been publishing now for several years many manuscripts that have first detailed how to make the beta cells and how to make the beta cells better, or use them to study patients, or use them to cure diabetes using various potentially clinical ways of doing it. One of the more recent things is a paper coming out next week is that we've actually recently been able to combine this technology with gene therapy to use the patient's own cells to cure diabetes in a preclinical model. So this is all part of the academic mission, but we're really doing this all with the focus on translation and recognizing that commercialization is key to this. And so the very early technologies that I developed in this space was used as the foundational intellectual property for a startup called Sima Therapeutics, which was recently purchased by Vertex Pharmaceuticals for $950 million. And so now we have a whole portfolio of new intellectual property, which includes the generation of the cells and uses of these cells in a lot of different contexts. And the big reason I wanted to speak at Venture Cafe today is that we're very interested in finding partners to help us with translating these academic findings over to something that could be actually beneficial for patients. And so the main two applications that we are interested in here are to leverage this platform to discover novel drugs. Again, we've worked out a lot of the conditions to be able to do the actual screening. And I'm not just talking about finding like new classes of secretagogues or other drugs that can increase in the secretion of endogenous beta cells. There's a lot of drugs on the market that can already do that. But I'm talking about novel compounds that can do things like prevent, slow down, or even reverse the loss of the native beta cells in patients with diabetes, or even expand the number of native beta cells inside of the patient to meet the metabolic needs of that patient. In addition, like I mentioned before, these cells can be used as an actual therapeutic themselves. And this is actually an area that I am collaborating with Dr. Zayed, the next speaker, on right now. But there's a major challenge in the field of being able to deliver these cells in such a way that they don't get attacked by the immune system, while also simultaneously ensuring the safety of patients and the efficacy of the transplanted cells. And so kind of our hope here is to be able to combine our academic mission and the new technologies we developed over the course of our academic mission with commercialization to be able to improve human health, to be able to have greatly expanded access of this sort of new technologies on the horizon to the millions of patients that could benefit from it, which would allow for continuous long-term control of blood sugar without the need of the patients actively monitoring it. And in the end, this would result in patients who are healthier and have lower risk of complications. So before I take questions, I wanted to quickly acknowledge the funding for my academic work, which is from the NIH, JDRF, and various centers at Washington University, and also give a shout out to my team who has come together to do all the work that we have been working on for the last several years and are continuing to persevere as we are facing the COVID-19 pandemic. So with that said, I am happy to open the floor up to questions. Thank you.
Hey, Jeff, it's Graf. Can you hear me?
Yes. Can't see you, but I can hear you.
Have you been in discussions with anyone yet who is on the device side about encapsulation?
Yes, we have active collaborations with academic labs working with encapsulation. We are talking with some companies who are actively working on that right now, but we don't have any active collaborations with any companies when it comes to the encapsulation problem.
Okay. One more question, if I may. Do you think ultimately higher likelihood of a product through encapsulation or through something that will make these cells tolerable to the host?
Well, that's always the hard question, right?
This is Winnie.
I simplified the talk down in order just to focus on encapsulations. All right, there's a lot of noise. There we go. Okay, I muted the person. I simplified it down to encapsulation because that's what I'm currently working with Dr. Zayed on. But I think it's an open question right now to determine if encapsulation is a solution or if other solutions are there. The paper that we have coming out next week in Science Translational Medicine presents a non-encapsulation solution to the problem, which is using the patient's own cells. And in this case, we were doing it with patients who had a monogenic form of diabetes. That's where the gene therapy came in to get cells, correct the gene variant, and then transplant it in there. When it comes to type 1 diabetes, which I know is your particular interest, I think that there's a lot of advances on the immune modulatory front that could perhaps be used as like a combination therapy with stem cell-derived beta cells. So either like tolerizing the immune system to prevent immune rejection of the cells, or to perhaps modify the cells themselves to make them invisible to the immune system, or to cloak them, in other words. But which one's the winner? I don't have a pony in the race when it comes to the winner. Whatever works, I'm very happy to use it with, and that's something for all the particularly from the companies I've been talking to. I've been challenging them to provide at least somewhat convincing data that their choice of it being, immune modulatory, immune cloaking, or encapsulation before we start working with them.
Right. Well, thanks, and congratulations on your work. For people who don't know it, you're really at the forefront worldwide. So that's awesome. Thank you.